In our last video, we submitted a taxonomic classification job using a reed sample that came from a study where they had collected samples from people who had had an orthotic replacement, like a hip or a knee replacement, and had had a bacterial infection. Now, these infections can be really serious because the bacteria can cluster around the new joint and they're sort of shielded from the immune response. So when something like this happens, they have to go in and remove the artificial joint, clear the infection, and then put it back in again or put in a new one. And so what they do when they removed it in this study was they collected back samples from it and then looked to see what bacteria were present in this sample. So we submitted a job from just one pair of reeds. They were aluminum reeds and they were the forward and reverse strands. And we submitted it to the taxonomic classification job. And now we're going to look at it. So how do you find the jobs that you've submitted in BV-BRC? Well, you can go down here and click on jobs here to get to the job table. You could go up into the workspace and click on your home workspace and navigate to it, or you could click on My Jobs here. So there are three paths that basically lead to the same place. You can see here that I have submitted a lot of jobs. And how do I find all those things? Well, if I can remember what it's called, I could look here and filter on something. Like if I had something like that, I could filter that and see what they were. Or I could go here and click on the down arrow next to all services and then click on taxonomic classification. And you can see here that I've run 824 jobs of that. So I'm going to click on that and it's showing them from the most recently submitted to the earliest submitted. Sometimes my names stretch on, so if I'm clever there, I can stretch it out and see, and this is the one I wanted to look at. We ran a microbiome analysis using the BV-BRC database. So let's click on that. And notice when I click on the row, the green action bar here gives me 3 different choices of things that I can do. One of the things that I could do is if I wanted to rerun this job or adjust it, I could click on the rerun button. And that will open a new tab and look at this. This is the run number that we submitted from SRA right here. So it's already preloaded for me. It tells me that it's a whole genome sequencing job, that I chose microbiome analysis in this database, filtering out the host reads at this confidence interval. Now, what it didn't reflect is that I said I wanted to save both the classified and unclassified reads. This is the folder. All I would need is a new output name for that. But we're not going to rerun the job right now. Another thing you can do is this other icon, Report Issue. If you click on that, this opens up this pop-up window, and this is a place where you can ask us questions about a particular job, and it will give us the information about that job. It's one thing if you go through the help section and I say my job failed, then there has to be this long dialogue back of what job, give me the job ID. What did you submit? Here, it's all here for the developer to look at. All you have to do is put in something like my job has been queued for 23 hours, which isn't true because this one ran, and then submit it. And that comes to the people at BV-BRC, and we look at that and try to figure out what's going on. So if you have a job that failed, we don't look at it unless you ask us to. So please use this. If you have any questions or your jobs are failing, let us know. But we're not going to do that. Today, we're going to look at the output from this job. So to do that, we click here on the view icon. So I click on that, and this is the landing page for a taxonomic classification job. This is showing the name of it and where it is in my workspace. It gives me the job ID number. It tells me how long it took to run. Remember when I submitted this job and I said it's a really big read file and it's going to take a long time to run? Obviously, I was wrong because it only took 8.5 minutes. So that's good to know. So let's say you've run a job and you haven't looked at it for a while, where it's getting time for a publication and you're trying to remember exactly what you did. You see this parameters section here? Let's click on this little arrow here, and that opens up the parameters box. And that's going to tell you, I mean, this thing can be a lifesaver. It will tell you everything that you did about this job. I often try to put it in the name because I run a lot of jobs and I want to be able to look at the name of the jobs to help me narrow down exactly what I want to look at. But it's all here. It tells me the sample that I submitted from the SRA. And look at this. When you do something like that, it goes in and gets the study title for that as well. It shows me the identifier that I submitted for that. It tells me what I want the job to be named, that it was a microbiome job. Remember that? It could have been species identification or microbiome, and we selected that. It says here that I wanted to save the classified sequences. If it says 1, that means yes, I wanted to save it. If it says 0, it means no, I didn't want to save it. It tells me the confidence interval I used, the database that I used, which was BV-BRC, or I could have used Kraken too, remember that. And then it says I wanted to save the unclassified sequences and the folder that I put it in. So everything is there. Oh, and one last thing, I told it to filter out the host reads, and my host in this instance was Homo sapiens. So all the information about what you've submitted is here. But the working parts of it are right here, and it's showing you all the data. Now we're going to handle this part of it in this video, but we're not going to touch on that, this particular folder, this mysterious folder, until the next video, because I like to kind of keep these things short. I don't want you guys to get bored. So let's talk about the sample key, CSV. CSV stands for comma separated values. So I click on that row, and then you notice the green action bar gives you a bunch of choices you can do from here. We want to view this. So I'm going to click on the view icon. And this is just telling me what samples were submitted. This was an easy job. It was just one set of read pairs. So we had the forward and the reverse strand, and these were the identifiers that I used. Now up here, this is the path in the workspace. Here is the job, and here is the folder. Let's click on the job here. And in this breadcrumb, it takes me back to the landing page for this job. Now we're going to do the MultiQC report. This is a very valuable thing that we've recently added. So I click on that, and I click on the view icon. And the MultiQC report brings in a lot of the information and collates it for you so that you can find things pretty quickly and look at things about the particular job that you've run. So on here, it's got the navigation that you can navigate to. There's a toolbox here where you can change things. And then here's the report itself. So let's start with the general statistics. The general statistics are just looking at the two read pairs with the host data and with the host data removed. And then it's also showing you the reports on those, both Bracken and Kraken. Now, if you're looking at this and you have some questions about what these columns mean, you can click on Configure Columns, and that brings a pop-up box that gives you definitions of everything and what is reported in this file. So it's pretty valuable to look at that. You notice that it's also showing up here, the top species in both bracken and kraken was Haemophilus influenzae, and bracken is a subdivision of kraken, and 96.7% of the reads were hitting Haemophilus influenzae there, and in kraken it was 66.2. For all the top 5 species, that's the top 5 altogether, and look, if I mouse over that at the top, it tells me who they are. Haemophilus influenzae, Streptococcus pneumoniae, Homo sapiens, Neisseria meningitis, and Trophiria whipplei. So in bracken, that was 99.6, and this was 68.3. This is information about the reads, and this is information about the taxonomy. The reads and the taxonomy. Then we can go down to top taxa, and once again we have a bracken report and a kraken report for that, and there's no mystery here because we've seen it up above that most of this is Haemophilus influenzae, and you can look at it by the actual number of counts. So if I mouse over that, it's showing me the number of counts in the second column of that pop-up window, the counts of reads, or the actual reads that are mapping to those individual organisms, the top 5. And by far, Haemophilus influenzae is the top. And if I go down here to this one, it's also showing that as well. So you can look at counts, or if I wanted to see the percentages, but I like looking at the counts. Now, another thing you can do that's kind of fun is you can march up the taxonomy and look at how it changes when you do this. So this is looking at the species level. If you do it at the genus level, they start looking a lot more similar, and you can see the blue are the Haemophilus, the black are the Streptococcus, the green are the Homos, Neisseria is peach, and Trophiria is the purple. And then we have other and classified. And this is where you can see that you're seeing some of the differences here in the kraken report, is it had some of the unclassified reads and other that are in here that were filtered out for the bracken. Duplication. So the definition of the duplication, it's bringing up minimizers. Kraken, if you read the Kraken paper, it talks all about k-mers, which are identifiers of a specific sequence that they're using uniquely for each taxonomy level. Kraken, too, in order to make things run more quickly, because this is a lot of data across all organisms are in here, they find it runs more quickly if they use something called minimizer or an m-mer. So we have k-mers and m-mers, and the k-mers are all unique, but the m-mers, the minimizers, a number of k-mers can be lumped in with one minimizer, but the minimizer should be, or the m-mer should be for specific for a particular taxon, whatever you're looking at. And so this is looking at the data from that perspective, and it's looking at who they are. And if you scroll down, once again, we're seeing our top hits. The big 5 are these guys. Then it gets into the FastQC report, which is the quality report. Now, I could go into this, but in the next video, we're going to be going into the FastQC reports so deeply. I don't want to do too much on this here, except to point out that this first one with the sequence counts, when you submit a number of different things, or let's say I have the forward and the reverse strand, and the forward strand showed that there were a large number of unique reads and duplicate reads, but let's say you could see that there was a great disparity here. Let's say you submitted a couple of samples and you could see that some looked better than the others, or that maybe the sequencer had failed on the other reads. This would be a place where you could look at it, see that there was a problem, and say to yourself, well, I'm not going to use this one in the publication. I'm going to either resequence this or throw it out and use something else instead, because it's of very poor quality. So you can look at its straight number of reads or the percentage of reads. We'll cover more about the FastQC report later on. So let's go back up, click on this. And now we can talk about diversity. There's a lot of talk about diversity, but we're talking about diversity here and looking at the microbiome report. Now, the Bracken reports does 5 different statistical tests looking at diversity, and it's trying to figure out if there are a lot of organisms in there or very few organisms. So you could look at the diversity statistics and determine that. And also if they are widely spread taxonomically or very narrowly spread. Let's first click on the alpha diversity.html. So I highlight that row. These two are essentially the same. The comma separated values in the HTML are essentially the same, but I like this one better and you'll see why. So let's click on that and highlight it. And here it shows the 5 different diversity tests that they run on every sample and the values for those. What I like about this is it gives me a definition for those things, because I don't know about you, I can't remember what the Shannon's diversity is, but if I look here, it's telling me how diverse the species in a given community are. A higher value indicates a greater number of species and the evenness of their abundance. And if only one species was assigned, the value would be 0. Well, this is 0.2. Remember when we looked at that MultiQC report? What was most of everything in this sample? It was mostly Haemophilus influenzae. So that's why we have a very low value here. So you can use all of these things. You can knock yourself out and explore all of the different diversity indices. But, you know, we have the visualization of that. So we're good. I think this explains it. And you can maybe even slip that into your publication if you want it to look like you were up on your statistical analyses. So the reviewer would think, oh, this person knows what they're doing. Okay, let's click back on the breadcrumb here. Breadcrumb like Hansel and Gretel leaving breadcrumbs to march their way. So the comma separated value, we highlight the row, click on the view icon, same thing. If there was a lot of stuff, you could use the filter to look for things, but pretty self-explanatory. Breadcrumb up. Now we're getting to the best part. This is like the cherry on top of the sundae. So it's the visualizations. So let's first click on the Sankey HTML. This is something new that we've added to BV-BRC, and it's really great. So I click on the view icon, and here, look at this thing. Isn't that beautiful? It's just perfect. It gives you a visualization of everything in that data. The MultiQC report was one thing, but this gives you the taxonomic spread right before your eyes in color. And we're primates, right? We like color and it helps us see things, and here it all is. So along the bottom, we have the taxonomy and the different levels: division, kingdom, phylum, class, order, family, genus, species. So you can see that some things, like this is a nice flow to Haemophilus influenzae. They're all coming out predominantly here, although there are a couple of other things that we can see here, parainfluenzae and hemolyticus. Some things you can't get to the species level. The Neisseria, well, it did get to meningitis, but there are some things that, like up here, that you see they couldn't get it any further than this. Now, one thing I really like about the Sankey plot is it shows me pretty easily the number of reads that are mapping to that taxon. So for Haemophilus influenzae, It is 1.24 megabytes of reads. I've been doing a lot of manipulations and looking at these things and trying to determine how many reads I need to get a pretty good genome out of a sample. And this one, I can get it. And we're going to do that in a later exercise. I would certainly try to bin out a genome of this and expect that to come out. This guy that has a total of 381 reads, I'm not expecting to get a genome of that. Okay, so this is our beautiful Sankey plot, the newest member of the BV-BRC Taxonomic Classification output. Let's go back and click on the breadcrumb again. And this time we want to look at the Krona plot. This is the one we've had for a long time. It's still a goodie, but you know, there's a big division in BV-BRC of the people who are Krona and who are Sankey plot. I personally like both of them, but some people take it very seriously. So let's click the view icon. I just like the way it pops up with this pretty little circle. And if I shrink it down a bit, you can begin to see that that's Haemophilus influenzae. But we want to look at the bigger picture here because it's showing you the same information that you saw in the Sankey plot in this circular form, and it's kind of fun to play with. For example, let's see, I wanted to find something that wasn't popping out to me, so I can use this to start looking, and it'll highlight it here to show me what I was typing and the information about it. Unlike the Sankey plot, this is giving me the percentages of the things. Over here on the right side tells me the total number of reads and things like that. But this is where the Krona plot gets fun. So example, if I click on this Terrabacteria group and double click on it, it's going to open it up and show me at that taxon level how things are mapping. Now look at this. This is Streptococcus pneumoniae, 75%. We know it's not 75% of the reads. No, when you click on something and dig into it, it's showing you the reads that are mapped to this particular group, to the Terrabacteria group, which are almost 23,000 reads. But of all the bacteria, it's a tiny part of it. It's just really tiny out of the whole thing. So this is a way to look at that. And if I want to go back to the norm, then you can see it all there. And also you can take a snapshot of this and put it in a publication, which we don't have that for the Sankey plot yet. All right, let's go back one more time. And now here it is. It's like the siren calling you. I know you want me to open it, but I'm not gonna open it in this video because I don't want this video to be too long. You're gonna have to join me in the next video, and you're gonna see that there are folders within folders and files and files. We're not done with taxonomic classification yet. There's a lot more fun ahead of us. So I'll see you in the next video.